Say hello to the people online.
Hello.
Yeah. Okay, you should really be back by now. Get a vaccine. Yeah, if you guys would get a vaccine, it doesn't matter if it's effective or not. Get the vaccine. Are they not giving it to everyone now?
It's only available for 18 years.
Yeah, 18, 16, somewhere in there. Oh my gosh.
So half of us can't.
Yeah. Well, if you could, I mean, I don't know if it's effective or not. That's an issue. We don't know if it's effective or not. Maybe good for 3 months, 6 months, maybe 5 years, 10 years. I don't know. But we don't have the data for it. But it'll keep you from getting quarantined if you'll do that. So go in and tell them, oh, I have an underlying condition. I need one. Okay? All right, so set up your page. Remember, you guys online, pause this if you need to. You guys who are absent, it's online. Get those notes. Now you do, right? Okay. Yeah, if you guys are missing your spiral, you might always look back there. If I find a spiral on the floor because people knock crap over, I'll just put it up on the cart. So if you're missing your spiral, just look around there. It'll be back there somewhere. Okay, so this is, what is today? 7/21. This is Unit 11, and it's DNA and protein synthesis. We're going to hit on the protein synthesis today. We're going to start on it. This is period 7. Okay, so our teak is B6C, and it's a short teak. We're going to write the whole thing. It's pretty short, but it's all uber complex. So it's really cool stuff, though. So B6C says that you're going to explain the purpose and process. There it is right there. Explain the purpose, which purpose is easy. Explain the purpose and process. It's the process that takes a lot of time. Purpose and process, and let me make this bigger a little bit so you can see it better. Purpose and process of transcription and translation. Using, here's the kicker, and we're going to have to make this stuff, using models of DNA and RNA. Obviously, they're not going to have models for you on the test. They're going to have pictures, that's about it. So we're going to do this using models of DNA and RNA. Okay, and here's the word I want you to put a box around. We're going to hit on transcription today. Do any of you guys know what the word transcription means? Transcribe, transcription. You are transcribing notes right now. What are you doing? You're writing them down. You're copying them, right? Transcription, the better you are at copying them, the better notes you have, the easier it'll be for you to study, the easier it'll be for you to answer questions when I ask them. So you copy them. The less mutations you have on your notes, the better they are. Yes? Okay, so copy them as I write them and you won't have that problem. So for this set of notes, this is going to be transcription. And I'm going to put in parentheses protein synthesis. Because transcription is one of the 2 steps of protein synthesis. So transcription and translation, they take protein synthesis. Protein synthesis, making proteins. That's what it means. What are your bodies mostly made of? There you go. Okay? So we're going to start with the overview because this is telling us explain the purpose. So we need kind of that overview or purpose. And when I say overview, we're not going to just focus on transcription. We're hitting all of it. So the overview or purpose of transcription, and I can't really abbreviate these because they'd abbreviate pretty much the same way. And translation. And that purpose is gene expression. I'm going to say it in 2 ways. Gene expression, the purpose of doing this is gene expression, otherwise known as making a protein. So when we talk about making proteins, if I can spell protein today, we're talking about expressing our genes. If you express your feelings for someone, you're showing those feelings for someone, right? You're telling them, you're showing them somehow. When we express our genes, they're showing up. Now, we see obvious traits like hair color, eye color, skin color, freckles, no freckles, things like that, right? Yeah, those are the typical traits or expressions of the genes that we think of. But it's everything. How big is your third toe on your left foot? That's a gene, that's an expression of that. Yeah. So when you look at these things, when we talk about transcription and translation, we're really focused on gene expression, which we do that by making proteins. The gene is expressed by making a protein. Whatever that gene says, that's the protein that's going to be made. So gene expression and protein synthesis are the same thing. And here is that central dogma again, that most important thing. So we start off with DNA. The DNA is copied. It doesn't go out of the nucleus. Your DNA has to stay safe inside that nucleus, right? You got to protect that stuff. That's really uber important. We make a copy of a section of it, and we do that by making RNA from the DNA. And then we actually translate the RNA into a different language, the language of the protein. So then we make a protein from that. What are proteins made of? Are nucleic acids and amino acids the same thing? No. Proteins are made of a chain of amino acids. Nucleic acids are made of a chain of those nucleotides, right? Sugar, phosphate, nitrogenous base. Sugar, phosphate, nitrogenous base. Those are a chain of nucleotides, right? So here, this is in one language, it's the nucleotide language, and then we change it to a different language, the language of the amino acid. So what happens is this piece, to start off with, going from DNA to RNA, is what we call transcription. And then going from RNA to protein is called translation. We're actually changing the language. It's no longer written in the language of the nucleotide, it's written in the language of the amino acids now. Okay, so now let's talk about that transcription. So this is just the overview right here. This is talking about the whole thing, right? Now let's focus on the transcription. So transcription means to copy or to write. Or write down. Let me just put write down on there. It makes me happier if I put write down. Makes more sense that way. The purpose of just the transcription, the end product, so I'm going to put it in two ways. The purpose of just the transcription part is to create a messenger RNA. And I'm going to write this, and you write it like this. It's a lowercase letter m, capital R, capital N, capital A. It's called mRNA. There are lots of different types of RNA. mRNA, rRNA, tRNA. Those are the ones we're going to talk about. There are other types of it, but you don't care. So the purpose of the first part of protein synthesis is just to transcribe. It's going to write it down, and it's going to write it down in the form of this RNA. And there are 4 steps here that we're going through. So here's the process. Here's the purpose. Here's the process. So 1, part of a DNA strand or ladder is unzipped. I'm going to use the word ladder here because it makes more sense to you, doesn't it? And when I say unzipped, do you understand what's going on? Yeah, you've got 2 halves of the ladder, it's going to split down the middle. It doesn't have to begin at the very start of the DNA, the whole thing. It can be, you know, you've had those zippers and they kind of unzip in the middle of it and it's a bad thing. That's not a bad thing here. It's going to unzip a little bit in the middle. It's going to open that little part of it here. So part of a DNA ladder is unzipped. What part? It's a gene. 1 complete section of DNA that has the instructions to make 1 protein. That's what unzips there. So it might be really long and the first part unzips, and as it goes down, it's going to unzip the rest of it and make a copy of it here. 2, so you've got to unzip it, right? You've got to open it up so we can make copies of it. 2, an mRNA is formed. How many sides does the RNA have to it? Remember, the DNA has two parts to the ladder, two sides to the ladder, and the RNA is like half the ladder. Just slice it down the middle here. So an mRNA is formed by making a complementary copy of one side of the DNA strand. So an mRNA is formed by making a, and I'm going to write this word down here, complementary copy of one side of the DNA strand. And that one side of the DNA strand that they're making a copy of, they call it the template. Do you guys, sometimes you hear words but you don't necessarily know what they mean. The template would be like, this is the copy I'm using. So, have you guys ever seen someone like sew something? Like when they've got a pattern, that real thin tissue paper, and they lay it out on the material and they cut out around it? Yes, no? Okay, that real thin tissue paper, that's the pattern or the template. So another word for template would be the pattern. You don't sew the paper when you're making something, you sew the material that you cut it out from, right? Yes, no? Yep. Okay. So an mRNA is formed by making a complementary copy of one side of the DNA strand. Okay, so the complementary means it's the other half. So if I look at my DNA ladder here, I've got one side, and here's the complementary side. So if I'm making a copy of this side, I popped it open. I'm not going to put a C there. What matches to the C? G. What matches to the A? T. But what if I'm making RNA? Will it be T? Will it be U? And then what matches to the G? So I would make the other half the complement of the strand, and that would be how I would make my mRNA. And then when I pulled the main mRNA out, that whole DNA ladder would pop back together. Okay, so step 3, uracil. Remember uracil replaces thymine. In the RNA. And we'll talk about what the mRNA means and all the other RNAs. We'll do that tomorrow when we do this. And then 4, one gene is copied at a time. Okay, it's more complex than that. But if we get this in our brains here, we're copying one gene. So you copy one whole gene, that mRNA is a copy of that one gene. Yes? So one gene is copied at a time. And then what's a gene? I've already written this down 3 separate times. Anybody remember? It's a piece of DNA that does what? That does what? Makes how many proteins? 1. It's a piece of DNA that has the instructions to make 1 protein. How many proteins? 1 specific protein. That's what that gene is. So it's a piece of DNA that has the information to make 1 protein. And it doesn't change. That's the only protein that gene is going to make. And then the last thing I want you to write down here is where I find that I have people that struggle with a couple of the words in here. So I'm going to write these down separately. And the first one is that word complementary. A good way for me to describe this is I can say that my husband is my complementary partner. He's not exactly like me. He's not an exact copy of me. He fits with me. He meshes with me. So when we talk about it, it's that opposite completing strand in the DNA. It's the one that makes the other half of the DNA complete. Not complementary like, oh, you look very nice today. Yeah, that's not what we're talking about. It's the other half to it. Think of puzzle pieces. One puzzle piece complements the other. They make a whole unit that way. And then the other word is template. If you're talking about a template, you're talking about a pattern. You could say, okay, I'm going to make $50 bills because that's how I roll. you would have to have a pattern to work from, wouldn't you? You'd want the best one to work as a template. You wouldn't make a copy and then use that copy to make another copy and then use that copy to make another copy, because that would degrade down the time. Have you ever gotten a paper from a teacher and oh my gosh, the copy is terrible, it's really nasty and pitiful? That's typically what happens. You have your original, which was beautiful, and then you make a copy of it, and maybe they pass it on or share it with someone else, and then they make a copy of the copy, and then you make a copy of that copy, and so on and so on and so on. That's how the copy gets degraded as time goes by. So the template means you have this pristine original, and we're not going to make a copy from a copy from a copy. We're going to go to the original, and we're going to get our copy there. When we want to make another of that protein, we go straight back to the original, and we make a new copy from the original template. Yes? Okay, flip over to page 21 in your packet. Oops, sorry, that didn't show up there. Okay, now, up here on the teak it said that you're going to use models of DNA and RNA. So this is the starting place for making your models for the DNA and the RNA.